Hi everyone, welcome back to Useful Genetics. This is Lecture 1C. We're going to be talking about how we represent DNA. Representation is really important for DNA because we can't actually see it. And this means we're very dependent on having conventions that everyone agrees on, where we know what we're talking about when we draw something to represent DNA. So genetics is necessarily and has always been about things we can't see. In classical genetics, when genes were first discovered, they were purely hypothetical. Nobody had any idea of their properties at all. We know a lot more now. We know all about the molecules, but they're still too small for us to see. So we can't just look at them. We can make very accurate representations of the molecules. These are molecularly correct drawings of DNA molecules. But in fact, such representations are not very useful. They're much too detailed unless you're a person who's physically studying the structure of DNA, which we're not. We'll find that much simpler representations are much more useful. For example, often we will represent DNA and chromosomes by simply drawing a line. That's about as simple as a representation as you can get. If we care about the orientation of the DNA, remember the business about 5' and 3' ends, we might use an arrow to mark the 3' end of the DNA. By convention, when we think about DNA, when we represent a single strand of DNA, we generally represent it with the 5' end on the left and the 3' end on the right. Just as there's a convention for text, the same sort of convention applies for DNA. If we need to think about both strands, we may draw both strands as two lines, and we may include arrowheads on the lines to remind us of their anti-parallel direction. If we want to really remind ourselves that these lines are DNA held together by base pairing, we can draw vertical lines connecting the DNA to indicate the base pairs. They don't have to be molecularly accurate. They're just there to remind us of what we're looking at. Very often what we care about in DNA is the sequence. So often we will write the sequence of the DNA. We won't bother drawing the DNA or making a line at all. We'll simply represent the sequence. Again, the sequence is by convention. We write the sequence of a single strand. Because the information is redundant, we don't need to write the information of both strands. We can always infer the information of the second strand from the information of the first strand. We always write the single strand from 5' end on the left, 3' end on the right. Sometimes there are circumstances when we actually want to consider both strands of the DNA. For instance, in Module 2, when we're talking about mutations, this will be important. And in those cases, we may draw the DNA, the sequences of both strands. In that case, the second strand is written in the opposite orientation, from 5' to 3', so the DNA can be shown correctly base paired. Now, here's an example of a very simple representation. You saw this in the previous slide, but it's easier now for you to interpret it. The two lines represent the two strands of DNA. With crosshatches indicating that they're base paired, and then they come apart and are single strands of DNA. The wavy line is representing RNA, and I've drawn it slightly thicker just to distinguish it from the DNA, and I've also drawn it kind of wavy. This helps us to remember that it's a different molecule, and I draw it wavy because RNA molecules are intrinsically more flexible, being single-stranded, than DNA molecules are. So this is RNA. I've drawn a protein that's acting on the DNA. In this particular case, it's an RNA polymerase, but it could be any protein. And I've simply represented it as an oval, a blob, with an arrow on it to indicate that it's moving. So all of that information is conveyed in this very simple set of lines on a page. Now, there's another level of representation that's also extremely inaccurate, and that's that we draw the DNA as if it was stretched out neatly, but it's not. DNA molecules are far, far longer than the cells they're in. The sum of the chromosomes in any one cell is about a meter long, whereas the cell is about 100,000th of a meter across. So the DNA has to be very tightly coiled to fit in the cell. And it's not just scrunched together at random, it's carefully packaged. So here's what we'd call naked DNA. This is rare in the cell, except at times of DNA, at places where the DNA is being directly copied. More commonly, almost all the DNA in the cell is initially compacted by being wound around proteins, producing a structure that's often described as a beads on a string. And then those beads with DNA wound around them are themselves wound around each other to make a thick fiber called the 30 nanometer fiber. You don't need to remember that. All of the inactive genes and the non-functional parts of the chromosomes are in this structure or in even more compacted forms in all of our cells, except when the cells are replicating. When the cells are actually dividing, the chromosomes need to be made even smaller so that the length of the chromosome is less than the width of the cell. And then those chromosomes are compacted even more by taking these fibers and again winding them up around each other. Now, here's a question for you. This is, again, as with the previous question, I'll show you the question, I'll introduce it, then you can answer it in the Coursera format before we'll come back and discuss it. So here's a drawing, a representation of DNA, representing the two strands of a double helix. What's wrong with this figure? What's wrong with this figure is that the two strands are not shown as being anti-parallel. The two arrows indicate that the two strands are going in the same direction, as if we had the 3' ends of both of them together and the 5' ends of both of them together. This is a structure that's physically not possible. The bases can't pair with each other when they're in the same orientation at all. Even if an A is opposite a T, they can't form a base pair when their strands are running in the same orientation. So what have we done? We've talked about different ways to represent DNA, why we need to have ways to represent DNA. Unlike people who study mice or flowers, we can't see it at all. So we have important conventions that we can represent DNA by something as simple as a line, but if we're drawing a line, it represents DNA running in the 5' to 3' direction. We use arrowheads to indicate directions. If we can write two strands, but we don't need to show two strands because the sequences are complementary. Now, coming up next in Lecture 1D, we're going to talk about the history of DNA, starting with the replication of DNA. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 1D, where we're going to talk about the history of DNA. We're going to start with DNA replication, a very short-term history of DNA, and then we're going to draw a parallel between the cell theory in biology and what we could call a DNA theory for the continuity of genetic information. Now, here's a diagram showing DNA replication with many of the components shown. This is much, much simpler than the molecularly correct drawing animations that you can find online. Here's a slightly simpler version. I'm going to draw for you a even more simple version, but first I want to just annotate this version with something that's left out to explain what's going on here. So the wiggly lines are DNA. Hang on, let's clarify that. So the curved lines are DNA, two strands of a double helix being replicated here, separating to form single strands that can then be copied by the process of DNA replication. What's not shown here is who's doing the work, and that's the enzyme DNA polymerase, which is actually a very complex factory of proteins. Which, if we drew it in, would completely obscure what was happening to the DNA. So DNA polymerase has bound to the DNA and has separated the strands and is then inserting new subunits in the single-stranded region, elongating the new DNA strands here, adding new bases, each base's identity determined by base pairing. Because of the polarity of DNA the fact that the strands are antiparallel, one new strand is being made in this direction, the other new strand has to be made in the opposite direction. But the same thing is happening at both positions. Now I'm going to do a simpler drawing of the same process. So, and I won't bother doing the helical structure, I'll just show the two strands of DNA, base paired, continuing off into the distance of miles of DNA, and coming apart under the influence of DNA polymerase. DNA polymerase is then synthesizing new DNA by inserting new subunits, checking the base pair for complementarity to the existing base. The same thing is happening on the other side, but in the opposite direction. The most important thing about this process is that it's directed by base pairing. So the base that's inserted here, this new red base, is identical, is paired with this blue base, and it is identical to the blue base that's present here at the same position. So if this blue base was an A and it was paired with a T here, this new red base will be a T. And the same at every position as the new DNA is synthesized. So the consequences of this for the relationship between the new molecules and the new two, what we call the daughter molecules, double-stranded DNAs, and the original parent molecule, is that the genetic information in the two daughter molecules is identical. So an A would be inserted here across from this T. The genetic information in each of these is identical to the information that was present when the two blue strands were originally paired with each other. Each daughter molecule consists of one new strand and one old strand, physically, but genetically, they are both identical to the original strand, original double-stranded DNA. Now, I want to think about this in the bigger context, and for a biologist, the bigger context is the evolutionary continuity of life ever since the origin of life. So this is the big tree, we call it. It shows the relationships of all of the organisms that are alive now, all the bacteria, the bacteria-like cells called archaea, and the eukaryote cells, which includes all the plants and animals. All of these organisms are descended from earlier organisms that in turn are descended from a common ancestor that's the ancestor of all life. Now, this picture is supplemented by something called the cell theory. This is a theory that was developed in around the 1850s, but when good microscopes became available and it was possible to see how cells divided. And at that time it was realized and popularized by a German named Virchow that all cells came from existing cells by cell division. Our bodies do not create new cells from scratch when they want them. Instead, all of the cells in our bodies arise by cell division from cells that are already present, all the way back to the original fertilized egg that formed when the egg and sperm fused. And the consequences of this realization is that this means that not just life, but that cells can be traced all the way back, that every cell of every bacterium and every cell of every archaean and of every eukaryote is a direct lineal descendant of cells that were present earlier in evolutionary time, all the way back to the very first cell. So cells aren't created from scratch. They all arise by division of existing cells. This is also true of DNA. Every DNA strand, as far as we know, every DNA strand originates by DNA replication using a pre-existing strand as a template. So this means that when we look at this picture, the big tree, it's also a tree showing the evolutionary continuity of DNA. All of the DNA in the bacteria, all the DNA in the archaea, all the DNA in eukaryotes, all the DNA in our bodies is direct lineal descendants of DNA that was present in earlier organisms, in earlier cells, all the way back to the very first DNA molecules at the origin of life. And this means that DNA, because DNA is also an informational molecule, unlike cells, DNA contains a lot of information about its evolutionary history. And we'll talk more about this later in this module. So what have we done? We've talked about DNA replication, about how each strand in an existing molecule acts as the template for creating a new strand that's complementary. And the result is that each new double-stranded DNA molecule has one old strand and one new strand physically, but genetically is identical to the parent molecule. DNA thus has evolutionary continuity. DNA isn't created from scratch. Every DNA molecule arose by replication of existing DNA. And this means that DNA's information is in some ways a record of evolutionary changes in DNA all the way back to the origin of life. And because DNA is the hereditary material that specifies all the properties of life, this makes it an enormously valuable resource for understanding our evolutionary history. We'll talk more about this at the last lectures in this module. Coming up next, we're going to talk about genes. We'll start talking about genes, first considering what is it that makes some of the DNA sequences in our genome be genes. I hope to see you there.